Okay, so our next speaker is an old friend, Alvin Luk, from Huida Therapeutics. And his title of his talk is Next Generation CRISPR Genome Editing to Combat Fatal Genetic Disease and Extend Lifespan.
Thank you. Okay, good morning. Thank you for the invitation again. I think last year I talked about most of them are mice. This time I'm going to talk about the human data of our two trials. And please go and visit two booths right at the entrance. One is from us in the F3. The other is Syndigo next door to us. It's on the F2. This is a disclaimer. For the disclosure, the two drugs that I'm going to talk about have not been approved. We're going to talk about the human data in a minute. First, I want to talk about the discovery of the 12-MAX and the Cas13. This is actually come from a precise platform that we actually look into the metagenomic database and align with different sequences, and we do a bunch of prediction for structure and the assembly of the phenotype. Then we do in vitro, in vivo assays, identify new nuclease, and then we again using AI and as well as the structural direct mutagenesis to do several mutagenesis. Then finally, we identify new nuclease that we're going to talk about here. They are all small, as we talked about yesterday from the keynote speaker. This is important. It fit into one single AAV. We have a bunch of other nucleases you can see from DNA editors to DNA base editor, as well as epigenetic regulators. From the RNA side, we also have an RNase editor and RNA base editor. This platform actually done by Professor Yan is right there. You can raise your hand. You can talk to him. I'm not a CRISPR person. Or you can talk to our CTO, TJ, right there. So anyway, so go into what's going on. I think we talk about, most of them we know about Cas9. The limitation of that is the size is too big. Then you can see the red dotted line is a single AAV. The capacity is over, so you cannot put it into one single AAV. Majority of the trial they're trying to do is using dual AAV. Of course, you can look for into a smaller nuclease like the 12F, but if you look down to the mammal, yes, you can go down to 400 amino acids, but the editing efficiency is suffer. Now, we do, using the precise platform, we identify DNA editor, DNA base editor, RNA editor, and RNA base editor. Want to draw your attention of the 12I, which is our 12-MAX, that we're going to discuss in a minute, as well as our Cas13, which is for our RNA editing. So when you compare to all the other companies with different editors, we see a lot of companies still working on, particularly on the DNA editor, from Cas8 to the Cas12. You also have base editor from Beam, again for the RNA editing, typically is RNAi, and then finally the RNA base editor from Scape. For us, we actually licensed to Syndigo last year, and then yesterday, Syndigo sub-licensed our Cas12-MAX to Vita for a cell therapy. For the 12-MAX, again, there's 4 important points to memorize or to remember. Small size, because we want to fit into one single AAV. High editing efficiency, and minimal of an off-target with a wide palm recognition. We did it in several different assays. You can see the editing efficiency for the LNP assays compared with the Cas9 is kind of equivalent. But if you look at the LNP, here's again, we actually show much better than the Cas9. For our in vivo, for the Huntington mice, but unfortunately we cannot do that in the Cas9. So by putting in a single AAV, but you can see that compared with the PBS, we are able to see that over 70% of the protein knockdown. We have 10 different models in mice and 1 monkey model for the Cas12-MAX. Again, you can visit the booth, both the Syndigo and us, as well as the poster today by Dow. I think he was right at the pole on the other side. For the Cas13, again, based on the precise platform, we are looking for small size, high editing efficiency, and also low bystand. When we compare with the RNAi versus with the wild-type Cas13, you can see the off-target issues versus when we engineer, we are much more precise. When we look into different models, such as the AMD with the VGFA, as well as the Angelman syndrome, you can see a very clear, specific knockdown of those genes. We have 5 mouse models and 1 monkey model for the Cas13. Now, so I'm going to switch into 2 different programs. One is the Cas12 for the DMD. Needless to say, this is a fatal neuromuscular disease affecting almost 200,000 patients. The gene is quite big, so normally with the traditional AAV using a microdystrophin that are already approved by the Disruptor, again, what can we do? I always think that using the traditional AAV gene therapy affecting the skeletal muscle cell, which is a dividing cell. If you look into the Disruptor phase 3 data at the year 4 and afterward, they all patients coming back down. But that makes sense because they are dividing cell. So in our approach, we are actually using a DNA editing. So we actually, in the DNA, the DMD gene, you actually see the stop codon that will actually have misprint on, particularly on the pre-mRNA. In the mRNA level, because of the stop codon, you actually have incomplete mRNA. Finally, you have the protein that is non-functional. In our case, we are putting into one single AAV with the Cas12 and with the guide RNA targeting to the exon 51 of the DMD. So this allows with exon 51 skipping and reframing. Because of exon 51, you can have a much better, bigger population of 14%. Our mechanism was clear. We can induce exon 51 skipping as well as reframing to restore the dystrophin production of the protein expression. Now we compare. I actually showed it last year that with the DMD mice. Actually, I'm not going to show you in here because since I taught last year, I want to compare this one with Vertex. Now, Vertex using the dual AAV with the Cas9. Look at the fold of difference. We are 6-fold lower using one single AAV. In their publication, in the 4-week versus with us, is comparable. So with this, we actually look into how about these HG302 with the Cas12 are they able to have a successful edit in the non-human primate? Again, you can see in the blue bar, they have all those product editing efficiency is acceptable for us. Then the next thing I will ask myself is, how about in functional assay in the mice? We actually do the rotor test as well as the grip. You can see that in the rotor test, it's much easier. You can see the non-significant difference between the wild type versus in our treated mice with the DMD. In the grips, about 16 weeks, you also see the non-significant difference between the wild type versus the treated mice with the HG302. Here is on the left side, it's treated with PBS. On the right side, it's treated with our HG302. You can see that the other mice, the other 2 mice on the left are falling down. Come on. You will see it. I turn off the sound. It's boom. You can know. So based on this, we go into our human trial. This is a first in human. In China, we call that an investigator-initiated trial. It's much less stringent. You can use a GMP-like material. You have to go to the ethics committee in the hospital. In this case, we actually run the trial between 4 to 8 years old with confirmed deletion of the exon 52 to 62, as well as 62 to 63. The patient are able to do the 10-meter walk independently. Because we are using AAV9, so we're also looking for no pre-existing antibody of AAV9 as well as the high fidelity Cas12 max. During the evaluation period, we look at the dystrophin expression. Unfortunately, as of this morning, 3 o'clock, I still haven't got my dystrophin data, but I do have the muscle function and some of the tests that you will see. Of course, a patient now is a standard of care for them using prednisone. Some of them are took it 1 year ahead before they joined our study. We were actually starting to lower and taper down of their standard therapy. In the long term, of course, we do a 5 years with the long-term stability and then patient outcome. Baseline, we dose two patients per each cohort, low dose and high dose. You can see ranging from 6 years old to around 8 years old. The weight is about 26 to 15 kilogram. Important, if you look down, is the NSAA, the North Star. Ground is that they still have a really good function. 6-minute walk is kind of range. One patient have almost 400 meters, the other only around 300 meters. This is more important for the safety, as we all know that Sarepta recently have a patient die after over 1 year with the AAV. But if you look, also there's another study that was using CRISPR. The first patient died, but again, this because of AAV-mediated toxicity. In our trial, we haven't seen these two patients have any SAE and DLT, no liver injury, and no thrombocytopenia. I think the more important is, as far as I know, most of the trials with the DMD with AAV gene therapy or CRISPR or base editing, they all have TMA. Some patients, it's manageable. Some patients went into the ICU for a couple of days and get out. We haven't seen that. The reason of why, because we are using much lower AAV. That's because the tool is much more effective and efficient. Then you look into the patient on the left. This is a patient will get up, and then he's still trying to have a little issues. Now, for the 91 day, this kid gained 5 kilograms, but he still can be able to get up. The fourth stair is a little difficult. He's been a lot of training. We're asking the kid to do one step on each stair. He won't able to do it until finally he learned it. He always have to do this and then step first. Now you look at, he got up. Unfortunately, on the other side, the doctor actually blocked it. You can see from the NSAA, we have 1 point improvement. This is clinical significant from the FDA. The 10-meter walk, actually, we don't see much difference between the 3 months after injection versus the baseline. However, in the 6-minute walk, we saw a 55-meter improvement, and the stairs, even though it's kind of look like the same, there was a half-minute improvement for that first patient. This is in a low dose of a low E13 VG per kilogram. So we are using a single AAV to deliver 12 max for the exons 51 skipping or reframing. We saw that we can restore functional dystrophin, comparable efficacy to Vertex at week 4, but we are 6-fold lower in the patient on the low dose. We dosed 2 patients. We haven't seen SAE, DLT, as well as the special interest, particularly in hepatic injury, thrombocytopenia, and TMA. Most of the AEs are grade 1 to 2, mild, transient, and resolved without intervention. For the efficacy, we saw the improvement, and the low dose is complete. We are going to the high dose. This program have been awarded for the rare disease pediatric designation and the orphan drug designation by from the US FDA. Besides doing this, we're also using different tools to look into different way of the DMD. We can do the editing, we can do the base editing, and now Professor Yan in his university are doing the gene activation that can cure all the DMD mutation. Again, today I just show you HG302 because this is a much larger population that we can reach in about 13%. But you can see we have a kind of strategy of working with DMD with different tools that we have. Now I'm going to switch into a different one using the Cas13. This is for RNA editing for MECP2 duplication syndrome. It is a fatal pediatric neurodevelopmental disorder. Most of the clinical symptoms such as intellectual disability, motor dysfunction, anxiety, as well as epilepsy. They have early death. Most of the patients will die or die before the age of 25. This is a sensitive gene, is MECP2. And what does that mean? It is loss of function you will have Rett syndrome. Many of the company, including Novartis, are using traditional AAV to really boost the MECP2. But FDA stopped moving forward for clinical is because they have no way to control, because if it's too much of MECP2, you get the other side, which is the MECP2 duplication syndrome. This company are using the promoter can moderate the amount of the expression of the MECP2. This is neurogen. Recently, the patient also died because they dosed all the way to 3 times e to the 15 VG. I'm going to show you in a minute. We haven't had that issues. So our strategy, again, using one single AAV to put into the Cas13, again with the guide RNA, that is focused on the MECP2 to knock down the mRNA. Therefore, we can actually reduce the protein. Because you're going to see that no matter how high we're going to go, it never go down to below the wild type level. We look into the mRNA level as well as the protein level. In the dose 5, you can see the no significant difference between the wild types versus our dose 5 treated mice with the MDS. More importantly, you're going to see that we also did not see it in the protein. So they are a very similar level with the wild type. Then look at in the neuron, this is expressing to the MECP2. If you draw into the attention of the wild type, you still see a little bit of the MECP2. This is because it is biological needed for neuronal functioning. For MDS mice, we actually see that the highly more expressed of the protein. And then again, treated with the MEC of HG204, you can see the decrease of the intensity. Looking into the brain, into the non-human primate, we can see that different region, on different region in the brain, and you can see the dose-dependent manner from low dose and the high dose. This suggests to us that this is actually appropriate going into the human trial. Then how about the function? This is also Yima asking why our MDS mice is on the top. Most of the wild-type animals should be on the top. That is because maybe again, that's a neuronal pathway that they are changed. Those mice are highly sensitive, highly hyper. So when they go into rotor, they actually really good on grabbing it. But however, if you look at the dose 4 and dose 5 treated with the into our MDS mice, you can see a dose-dependent manner. Again, on the week 8 and week 16, you continue to see the non-significant difference between the treated mice versus the wild type, suggesting that it's back to the normal level. One other thing for the Rett syndrome in publication, you will see that they have the mice die very quickly. In our one, the median life expectancy is about 155 days. After treated with the HG204, in the yellow stuff, because we have to sacrifice 5 mice, you can see the median range of lifespan is 226 days, but we do have mice pass over 300 days. Remember, I told you about one of the clinical symptoms for the patient is anxiety. So how can we test it in mice? In the vertical, you're going to see a wall. In the horizontal, it's an open field. You can see the wild type have no issue going to the open field, while the transgenic transgenic MDS mice, you can see they are not likely want to go into the in the horizontal. Versus after the treatment with the HG204, again, we can see that they can go to horizontal or the vertical. That means they can go to the open arm. Now I'm going to switch into the patient. Before I get into data, I need to talk about some of those pediatric developmental stage. At the beginning, most of the patients will have a misdiagnosis of those patients because they thought that the kid will have the delay of global development. However, as the time progresses, they will notice that it's low muscle tone, the head control, they cannot do it. They don't know how to do the feeding, or particularly with the bottles. By the time when they call the developmental regression, this is another state that they're starting to see that they do not know how to speak. They have a language communication. Fine motor will be another one. pretty much about the finger. In a minute, I will explain. And then, as early, some of the patient will have onset of epilepsy. Then you kind of maintain stable, but it's continued to decline. You can see the severe intellectual disability, as well as persistent communication defect. You will see a lot of respiratory infection. And finally, the patient will have the spine will curve, have a problem of walking. This is the gross motor. They will not do eye contact with you. Finally, they will end up in wheelchair, as well as the life die before the age of 25. I want to draw your attention in the developmental regression here. That is the global impression scale. That is 7 domains. I want to draw into attention of the 3 most important domains, the hand function. In the hand function, mostly you learn it, but you likely, if you're a patient, you will lose as you grow old. Gross motor, the advanced skill, you will never learn. And then for communication, you do not know how to express your emotion. So in the first column, the second column you see on the early onset, those are the skills that they learn when they are between the half years old to 1.5 years old. The grabbing is like the baby when they have the finger put into the palm. Then you can also call pincer, using the thumb to the index. They know how to do that. You can see how much they learn, and then sitting as well as walking. On the first stage of the regression, those are majority of those they are losing. Those are the percentage they lost, particularly on the grips as well as holding the bottle. Now they do know how to blah blah blah, but they cannot speak even single word. Now for the gross motor, they still retain. This is good, but you will see what happened to the video later on. Then the last part is those are the skills they never learn, such as using the fork or using the utensil to eat. They never learn how to climb up the stairs and go down, and they never express emotion, and they never know how to wave bye. So in this our trial, again, is investigation initiated trial with the first in human. We're looking for 2 to 17 years old with the confirmed MECP2 duplication syndrome. We do not want to deal with MECP2 triplication. One, they have stable seizures or have no seizure within 2 months before screening. Again, no pre-existing antibody with the AAV9 or the HiCap13. We do a log of scale because of the behavior and motors. Earlier I talked about the global impression scale. We also have Peabody and Griffin. Those are all developmental scales. We also have intellectual scale because I remember they have intellectual disability. They also have epilepsy, so we have ECG. We are trying to do a biomarker such as the GTF11. Long-term, again, durability and long-term safety. This group of patients, they are not going to have proteasome very early, typically a couple days before the infusion, as well as sirolimus, just to calm down the infection as well as inflammation. Low dose, again, I put it in 1E14 because the patient who died in the other study with the traditional AAV was in 3E15. Now they come back down to 1E15, but still we are tenfold lower than them. You can see the score, particularly in the function, language. Most of the patients we want to treat between 4 to 6. What does that mean? Here you go. The scoring between the definition, we want to focus mainly on the moderate to severe. You can see that most of the patients have the problem is the language, ambulation like walking, as well as hand use. So what's the stat mean of those 4 to 6? Here you go. The 4 earlier I showed you, that was the 4 is on the hand. So you have limited hand use. They do not know how to grab, and they all grabs. They will always dropping objects. For walking, they're mostly severe because they need assistance. You can see a patient later on in the video, the body like this, cannot walk, cannot sit, cannot talk. You will see that a doctor asks him to do things, he will not talk, and he will not do eye contact. So in terms of safety, again, we haven't seen any SAE and drug-related SAE and then the DLT. In this population, again, I'm using TMA because this is a well-known AAV-mediated toxicity. We haven't seen it yet. Autoimmune encephalitis is very well known for this population for the infection into the brain. So this is the first patient of 9 years old. Now, hand function before, my team say forget about all those words, just do the drawing. But okay, I want to show you that the kid will not know how to eat, so the parent have to feed them. Always dropping object. You can see that in the hand function. Briefly, he can hold the object and drop. And then finally, he cannot express emotion. 3 months after that, you can see in the video, he can reach out and do things. He does talk, he cry. This is the thing that the parent call us when ask the kid, come to eat. He recognize, he know what's going on. Go to the bathroom. Now, how many of you have kids or have been a kid? Think about that when the mother have to go to work, you cry. This boy know how to cry, but do not understand emotion, do not understand upset. So the mother call after almost 3 weeks. They have a problem now because he cannot leave home. Because every time go to work, the kid cry and get really upset. Now, I think in the left side, you're going to see, the doctor asked, throw the ball like this way. He won't understand. This is a 9 years old with only 11 months IQ. Now, after the injection, you can say, he said, give me the ball. Here you go, he don't understand what's going on. I told you again, the walking, you can see he needs 2 hands, the way that's how he walk. This is in the baseline. After injection, you see it's only 1 hand of a walking. A much better posture and much easier to do the gait. Finally, she doesn't know what to do with the stabbing. He also do not want to look at the doctor, no eye contact. You will see on the far side after 3 months, you can see he reach out. He know how to stack it up. Then, of course, we also look into other behavior. It's all have some improvement. So finally, this is the last 2 slides. I will be done. So we have AAV delivered with the Cas13 again. Again, we show you that safety. We also see improvement of the motors down here, sorry. And then this one, actually low dose is complete. We go to the high dose. This program is actually got RPDD and ODD from FDA, as well as EMA. So by conclusion, I want to tell you that we have the discovery of the 12 Max and 13Y, compact, small size, high fidelity, and efficiency. Both of them will show you the safety and efficacy of the 12 Max and the 13. And finally, this 302 will be in a President Symposium at the upcoming ASGCT 2025. But we also get ODD, RPDD, and that's at my acknowledgement, including our team with Syndigo here.